All right, ladies and gentlemen, we are on our final lecture for the semester. I know, it's a sad day. But today we will be going over the male reproductive system. To begin, think about what some of the functions of the male reproductive system might be. Like the female reproductive system, the most important function is to produce gametes. In this case, males produce sperm. On average, each ejaculation contains about 500 million sperm. And when you think about it, only one of those will fertilize the egg. There are many myths out there about fertility and conception. Sexual positions, the types of underwear men wear, or specific foods do not have an impact on getting pregnant, but your age, weight, and lifestyle certainly do. The male reproductive system acts as a transport system for those sperm, and the main hormone produced is testosterone. There are quite a few structures you will need to know for the male system. To orient us to the mid-sagittal cross-section, let's start with a few landmarks. Here we have the bladder, which you learned about in the urinary system. A blast from the 201 past is the pubic symphysis. And posteriorly, we have the rectum, which you learned about in the digestive system. Since creating spermatozoa is what this system does, we'll start at the source and follow their path to the external environment. Inferiorly, we have the scrotum, which is a sac of skin that contains the testes. Inside of the testicles is where sperm develop, and we'll learn about spermatogenesis in a few slides. The sperm travel from the testes into the epididymis, where they mature and are stored until ejaculation. Once ejaculation occurs, the sperm travel through a muscular tube called the ductus deferens, or also known as the vas deferens. They arrive at the ampulla. Next, the sperm pass by the seminal vesicles, which add various compounds to make semen. The sperm and semen enter the ejaculatory duct, which passes through the prostate gland. Here, the prostate gland adds more compounds to the semen, and it joins at the prostatic urethra. Finally, it passes the bulbourethral gland, which adds more fluid to the semen, and continues along the urethra. This is a shared pathway for urine and semen, so the secretions from the bulbourethral gland help neutralize any acidity from urine. You can see here that the root of the penis is surrounded by adipose tissue in yellow, and it is connected to the underlying fascia. It is also part of the penis, which no one can see, so it technically adds length to the overall size of a penis. The penis itself is composed of the corpus spongiosum, which surrounds the urethra, and the corpora cavernosa. The shaft of the penis, the external part of the penis, the end of the penis is called the glans, and it is surrounded by the prepuce, or the foreskin. This image shows the World Health Organization's estimates of circumcision from 2007. There are many religious and cultural beliefs for circumcision, but from a health standpoint, removing the foreskin can prevent disease from buildup under the skin called schmegma, and data shows that it can reduce, not prevent, the transmission of some sexually transmitted diseases. Here you can see an image of a cadaver that has a bisected penis, and highlighted in orange is the spermatic cord. The spermatic cord is surrounded by connective tissue and contains the cremaster muscle, the testicular artery, veins, and nerve, and the ductus deferens. Let's take a closer look at these structures and how it all works. Again, we'll start in the testes and work our way out towards the external environment. Remember, the testes are an endocrine gland, and they produce testosterone, but they also have an exocrine function, which is to produce and secrete gametes. In most males, the left testicle hangs a bit lower in the scrotum, which reduces compression between the two testicles. The testicles are surrounded by a connective tissue sheath called the tunica albuginea. If you remember, we saw this same connective tissue layer surrounding the ovaries, because these organs are homologous. It's during embryonic development that genes regulate and control their differentiation. The testicles are surrounded by two muscles, the cremaster muscle, which elevates or drops the testicles, and the dartus muscle, which causes wrinkling in the scrotum and reduces surface area to reduce heat loss. Together, these muscles work to bring the testicles closer to or farther away from the body and help to regulate the temperature of the testicles. When it's cold, the muscles contract and bring the testicles closer to the body to warm them up, and when it's warm, they relax so the testes can be farther away from the body in order to cool down. Another method used for thermoregulation, or managing the temperature of the testicles, is through a countercurrent heat exchanger called the pampiniform plexus. It is a network of veins that surround the testicular artery and helps to cool blood flowing through the artery to the testicles. You may be asking, why would the body try to cool the testicles? Well, if you look closely, the blood leaving the body through the testicular artery is about 37 degrees Celsius or 98.6 degrees Fahrenheit. As blood flows through the artery towards the testicles, it loses heat to the cooler blood in the pampiniform plexus. So by the time it reaches the testicles, it is about 35 degrees Celsius or 95 degrees Fahrenheit. Sperm fully mature at 35 degrees Celsius, which is why the body is working so hard to regulate the temperature of the testicles. The title of this slide is hyperlinked to a great visual resource to help add clarification if you should need it. This slide highlights the structures you need to be familiar with and should be able to find in the dissection for this lab. Towards the top of this image is the spermatic cord. At the bottom, you can see the scrotum, which has been cut and peeled back so we can see the internal structures. In the center is the testicle, and as we'll see shortly, it is connected to the head of the epididymis. The head of the epididymis leads inferiorly to the tail of the epididymis, and finally up through the ductus deferens. To connect the importance of understanding our bodies, I put in some relevant health information. In 2018, approximately 9,300 new cases of testicular cancer will be diagnosed, and 400 deaths will result. Therefore, it's important for males to check regularly and to see a doctor if you have any concerns. You can give yourself regular exams every time you take a shower, and you'll want to do this when the skin of the scrotum is warm and loose. Start with one testicle and firmly but gently examine the surface with your fingers, looking for any irregularities or changes from previous exams. And then repeat with the other testicle. Now we're going to take an even closer look at the testes to see where the sperm are produced. Each testicle contains coils of small tubules called seminiferous tubules. An average male can have up to a half mile of these tubes in each testicle. All of these tubes converge on the ready testes, which leads to the head of the epididymis. Here, sperm are stored for about 2 to 3 months where they mature in order to be able to swim and fertilize the eggs. Sperm then make their way through the body and into the tail where they further mature and remain until ejaculation occurs. Upon ejaculation, the sperm make their way into the ductus deferens and are propelled by smooth muscle. Here we have a cross-section through a seminiferous tubule. This is the site of spermatogenesis, in other words, where sperm are formed. The tubules are surrounded by connective tissue to prevent them from shifting around, and in the space between the tubules, we find the interstitial cells. These cells are responsible for producing testosterone. The spermatids develop inside of the seminiferous tubules and receive nutrition for their development from the sustentacular cells. As the spermatids grow, they start their development superficially and move deeper towards the lumen of the seminiferous tubule. To show you a more detailed perspective, we can see here again that we have a cross-section of a seminiferous tubule. For reference, here's that superficial connective tissue layer. Spermatogenesis starts here with the spermatogonium. As it undergoes meiotic divisions, it moves towards the lumen and receives nutrition from the sustentacular cells. These cells also produce ABP, or androgen-binding protein, which helps concentrate testosterone in the seminiferous tubules to promote sperm development. The junctions between the sustentacular cells are called tight junctions, which prevents the body from attacking the germ cells. Over 25,000 sperm are released each and every minute during this process. Along the way, the developing cells lose most of their cytoplasm and finish meiosis, creating genetically different haploid spermatozoa. Under normal development, sperm have a few features you will need to know. Here you can see the head with an acrosome cap. The acrosome contains digestive enzymes important in breaking through the corona radiata of the oocyte to allow for fertilization. The midpiece has lots of mitochondria to make ATP for the flagella or the tail, which helps propel the sperm on its journey to the egg. Abnormalities do occur among the developing sperm, and you can see some examples here. Sperm morphology results are reported as the percentage of sperm that appear normal when semen is viewed under a microscope. These abnormalities may affect the ability of the sperm to reach and penetrate an egg, but having a large percentage of abnormal sperm isn't unusual. Abnormal sperm also do not indicate high or low fertility. Semen analyses also include volume, total sperm count, sperm concentration, the percentage alive, and their movement. Men with low semen analysis values can still have kids. It just might take a little bit longer. If you follow the hyperlink in this slide, it will take you to a video of a new sperm bot that was designed to help move individual sperm to the egg in order to reduce the high cost of in vitro fertilization. Remember, sperm develop in the seminiferous tubules of the testes and are stored, modified, and mature in the epididymis. Next, they move into the ductus deferens during an ejaculation and are propelled by contractions of smooth muscle called peristalsis, where they pass into the ampulla of the ductus deferens. Then they pass our first accessory gland called the seminal vesicle, which contributes about 60% of the components of semen. Semen is the fluid portion that suspends the sperm and contains nutrients, clotting proteins, and other substances important in promoting fertilization. This fluid passes into the ejaculatory duct, through the prostate gland, which adds more components to the semen, about 30%. The fluid moves into the prostatic urethra, past the bulbourethral gland, which adds a little bit more fluid, and finally into the penile urethra and out of the penis. According to studies performed by the Kinsey Institute, sperm can actually travel a whopping 25 to 28 miles per hour. Although, in most studies, it just dribbles out. But once sperm enter a vagina, they slow way down to about 6 inches in 5 minutes. Once they are in the vagina, a biochemical maturation process called capacitation occurs. This prepares the acrosome and stimulates the motility of the sperm. Remember, semen and sperm are different. Sperm carry the DNA, and semen is the fluid that suspends the sperm. Each ejaculate contains about 2 to 5 mils of semen, which is created by the accessory glands we already mentioned. The seminal vesicle secretes carbohydrates and citrates for energy, prostaglandins that help thin the cervical mucus and create microcontractions to help the sperm move, as well as praseminogelin. Praseminogelin makes semen sticky, so they adhere to the vaginal wall and canal and stick around longer. The prostate gland secretes a buffering fluid to reduce the acidic environment of the vagina, a clotting enzyme, and serin protease. This compound activates after about 20 to 30 minutes and breaks down the stickiness of the semen so that the sperm are able to swim more easily. And last is the bulbourethral gland. This gland secretes a bit of lubrication just before the sperm and semen travel into the penile urethra. This not only makes their movement easier, but it also helps to reduce the acidic environment from any residual urine in the urethra. The penis is made up of multiple parts. Here you can see the bulb and the crus, which together make up the root. The shaft and the glands or the head of the penis. In this image, the foreskin has been removed. If we look at a cross-section of the penis, we can see there are 2 corpora cavernosa, which are separated here by a median septum. They surround deep arteries, and inferiorly is 1 corpus spongiosum. That surrounds the urethra. During sexual arousal, blood flow to the penis increases and it fills the corpora cavernosa. As this tissue engorges with blood, it expands and closes off the dorsal vein, reducing the flow of blood out of the penis, keeping it erect. As I mentioned earlier, the urethra, the only channel for sperm and semen to pass through, is located inside the corpus spongiosum. The spongiosum absorbs the expansion during the erection and keeps the urethra open to allow semen and sperm to pass through. In order to promote healthy erections, good blood flow is required. Stress, smoking, and age can reduce the blood flow or reduce the elasticity of the arteries, which can affect erections. This is commonly called erectile dysfunction, and it's been estimated that 60% of doctors' visits related to ED are stress-related issues. In case you are wondering, in a study of 80 men, researchers found increases from flaccid to erect lengths ranged widely, from less than a quarter inch to 3.5 inches longer. The average length of a male's penis was 8.8 centimeters, or 3.5 inches flaccid, and 12.9 centimeters, or 5.1 inches erect. In labs this week, you will be looking at a primate penis in cross-section. You will have to move the slide around to get a good perspective because not all structures are visible at the same time. Here you can see the two corpora cavernosa separated by the median septum. And here are the two deep arteries. Sometimes students get thrown off when looking at these under the microscope because they aren't the same size or located right in the middle. There is variation, and that's okay. The urethra and corpus spongiosum would be lower in your field of view on this slide. Briefly, for those of you who may have had, or know someone that has had a vasectomy, this illustration shows how that happens. Incisions are made in the scrotum to access the ductus deferens. The ductus deferens are then cut or blocked off. This way, no sperm can get by, but semen will still be produced and released from the accessory glands. Another bit of health information you should be aware of is prostate cancer. It is the second leading cause of cancer in males. In 2018, it is expected that about 165,000 new cases will be diagnosed and about 30,000 men will die from it. It is recommended that you talk with your doctor if you or someone you know has trouble urinating, has a reduced flow, has blood in the urine, pain in the pelvic regions, or even erectile dysfunction. Your doctor may want to check to see if your prostate is inflamed or swollen, and the easiest access point is through the rectum. The hyperlink in the title will take you to the American Cancer Society for more information about prostate cancer. This week in lab, you will need to finish your lab notebooks and turn them in, but be sure to view the slides of the testes and penis and complete the bull testicle dissection. It is also hyperlinked here to a video if you need to review or can't dissect due to various religious or cultural beliefs. Well, that's it for 202 lectures this semester. Next week, you can pick up your notebooks from your lab TA, and all week long we will be having open labs. You can come into any TA's open labs and stay for as long as you would like. The practical will be the following week, and your lab TA will give you more instructions in lab. Start studying now, as this practical exam is very challenging. Thanks for tuning in to the online screencast. I hope you enjoyed learning about human anatomy, and I wish you the best of luck in your academic and professional careers wherever they may take you.